Hello guys, today we're going to discuss the genera of Enterobacteriaceae. First, what is the general characteristics of this genera? First, most medically important of gram-negative bacilli. Most species are normal flora of the GI tract, except for Salmonella, Shigella, and Yersinia, because this microorganism can cause clinical significance to an individual. Also, major cause of nosocomial infection. So this genera of the microorganism causes diseases, includes UTIs, gastroenteritis, septicemia, food poisoning, wound infection, peritonitis, pneumonia, and meningitis. So this is the general characteristics also of the Enterobacteriaceae. Gram-negative microorganism, facultative and aerobe, all reduces to nitrate to nitrite except for Pantuwea agglomerans. Also, all Enterobacteriaceae are catalase positive except for Shigella dysenteriae and Plesiomonas because these microorganisms are catalase negative. Also, Enterobacteriaceae is cytochrome oxidase negative or tinatawag natin na taxo-N. Also, Enterobacteriaceae is aerogenic. They produce gas, except for Shigella. Kasi guys, sa previous lesson natin, nabanggit na natin that Shigella, majority ng kanyang biochemical tests are negative. Again, majority ng biochemical tests ni Shigella are negative. Tatandaan, can be differentiated based on the lactose fermentation of the microorganism. And tandaan natin also that all Enterobacteriaceae microorganism can ferment glucose. Next, what is the four serological characteristics of Enterobacteriaceae? So we have the O antigen, K antigen, H antigen, and the V antigen. So this O antigen, also known as the somatic antigen, a cell wall antigen that is heat stable. Next, we have the K antigen or the envelope antigen. So this is also the capsular antigen or the heat labile. So ano ginagawa ng envelope antigen? It protects the microorganisms. So anong mga microorganism na may envelope antigen? So we have the Klebsiella, Salmonella, and Escherichia coli or E. coli. So next, we have the H antigen or the flagellar antigen. So this is a heat labile antigen. So we all know that the flagellar or the flagella use of the microorganism to move or movement para maging motile sila. Next, we have the V antigen. So tanda natin guys that V antigen commonly seen in Salmonella typhi. Unique characteristic siya ni Salmonella typhi. Also, V antigen is heat labile. So we need to discuss the rapid lactose fermenters microorganism. So we have the EKE. So tatanda natin guys, rapid lactose fermenters, EKE. So kapag nabasa natin guys yung RLF, that is rapid lactose fermenters. Pag sinabi natin RLF, ito yung microorganism na mabilis sila mag-ferment ng lactose. Ano yung mga enzyme na meron sa ating mga lactose fermenters microorganisms? So we have the beta-galactosidase and lactose permease. Ito yung mga enzyme na pwede natin makita sa rapid lactose fermenters na microorganism. So example of this microorganism is Escherichia coli, Klebsiella, and Enterobacter. Tandaan natin yung mnemonics, rapid lactose fermenters, EKE, E. coli, Klebsiella, Enterobacter. Next, we have the late lactose fermenters or LLF. Kung mapapansin nyo, dito we have the beta-galactosidase and lactose permease sa RLF. Kay LLF naman, wala na si lactose permease. Meron na lang si beta-galactosidase. So, sino yung mga microorganisms na under ng LLF? So, we have C. shai, we have Citrobacter, Salmonella, Shigella, Serratia, Hafnia, and Yersinia. So, tandaan natin yung ating mnemonics, C. shai. Next, we have Pseudomonas species. So in TSI, the result of Pseudomonas is alkaline. So, balikan nyo lang guys yung topic natin regarding sa culture media. So, hindi ko na diniscuss yung mga TSI, LIA, kasi na-discuss ko na siya. So, you need to back sa topic na yun. So, next. So, what is the microorganism that is urease fermenters? So, we have rapid urease fermenters, and we have the slow urease fermenters. So, ano yung mga microorganism na under ng rapid urease fermenters? We have ppm, Proteus, Providencia, and Morganella. Tatandaan, Proteus, Providencia, and Morganella. So, under naman daw ng slow urease fermenters, we have C. keys, Citrobacter, Klebsiella, Enterobacter, except for Enterobacter gergoviae, Yersinia, and Serratia. Pero guys, kapag tinanong sa board exam, which of the which of the following microorganism can ferment urease? So, hindi naman sinabi kung rapid or slow. So, lahat doon ng microorganism can ferment urease. Tatandaan natin, ppm sikis can ferment urease. Tatandaan, wag malilito. So, next, what microorganisms are non-motile at 37 degrees Celsius? We have Sky, Shigella, Klebsiella, and Yersinia. Tatandaan. Next, we have microorganisms that under Enterobacteriaceae that can produce pigments. So, red pigment produces by Serratia marcescens. So, ito yung tinatawag natin na prodioxin. Prodioxin. Ito yung responsible kaya nagkakaroon ng red pigment si Serratia marcescens. And Panduea and Enterobacter sakazaki produces yellow pigment. So this is the example of Serratia marcescens and Enterobacter sakazaki. So we need to discuss the each microorganisms that under the Enterobacteriaceae. First, we have Escherichia coli or E. coli. So the in-vivo reaction for Escherichia coli is positive, negative. So I for indole, M for methyl red, V for Vogt-Proskauer, and C for citrate. Tatandaan natin yung INVIC. So, Escherichia coli other name is colon bacillus or golden bacillus. Ba't kaya siya tinawag na colon bacillus? Kasi guys, Escherichia coli or E. coli are commonly found in our colon. Normal flora siya ng ating colon or intestine. So, the TSI result for Escherichia coli is A over A or acid over acid, and of course, H2S negative. Kasi nga, na-discuss natin that microorganism that produces hydrogen sulfide yung ating space. Di ba? May mnemonics tayong space. Tatandaan natin yun. So next, in Maconchi agar, Escherichia coli produces pink or red colonies. In sheep's blood agar, produces beta hemolytic. And na-discuss na natin previously sa ating culture media that Escherichia coli produces green metallic chain colonies in EMB, indole positive, ONPG positive, methyl red positive, Vogt-Proskauer negative, citrate negative, and urease negative. So, di ba, natin kanina, yung mga mnemonics under urease. So we have different strains of Escherichia coli. So first we have ETEC or the enterotoxigenic E. coli. So ETEC causes traveler's diarrhea or transient diarrhea, produces cholera-like toxin. Tatandaan natin, cholera-like toxin. And the stool sample is watery. In Brazil, they called it turista. In South America, Montezuma's revenge. In Japan, Tokyo two-step. And India, Delhi Belly. Tatandaan natin itong mga to. Next, we have the epic enteropathogenic E. coli. So, this is the infantile diarrhea and it has strain of E. coli 0111. Tatandaan natin yung strain. And the stool sample is watery with mucus but without blood. Tatandaan natin, watery with mucus but without blood. Next, we have the AIEC or the Entero-invasive E. coli. So it produces Shigella-like diarrhea, watery with blood, with pus or WBC, and of course, mucus in our stool sample. Next, we have the EAEC or the Entero-aggregative E. coli, causes acute and chronic diarrhea, produces adherent biofilm, and the stool sample is watery with mucus only. Next, we have the EHEC or enterohemorrhagic E. coli. It produces Shiga-like toxin. So we have VTEC, the virotoxigenic E. coli, produces virotoxin, and the stool sample is watery with excessive blood but without WBC. Tatandaan natin, watery with excessive blood pero walang pus or WBC. And EHEC causes HUS or the hemolytic uremic syndrome. So this HUS is a group of blood disorder characterized by low red blood cells, acute kidney failure, and low platelets. Kasi nga, bakit mababa daw ang kanyang red blood cells? Kasi nga, excessive blood, di ba? In-excrete sa stool yung ating blood. So, normally, mababa ang ating red blood cells. So, we have the strain under EHEC. We have the 0157H7, which is the common type. So, this strain of E. coli is non-sorbitol fermenter. So, tatanda natin, this strain of E. coli is non-sorbitol fermenter. Next, we have the anaerogenic E. coli or the E. coli inactive. So, this is alkalescence dispar, does not produce gas during the fermentation. Kasi nga, sabi nga natin that all microorganisms that under Enterobacteriaceae can produce gas except for Shigella. Next, we have Klebsiella. The Invik reaction for Klebsiella is negative, negative, positive, positive. So the identifying characteristics of Klebsiella in TSI is A over A and produces gas. So in McConkey produces colonies, but the colonies are mucoid because of the capsule production. So H2S negative, metal red negative, non-motile, indole negative except for Klebsiella oxytoca and Klebsiella ornithinolytica because they are indole positive, VP or Vogue cover positive, citrate positive, and of course lactose positive. So this is the example of Klebsiella under McConkey agar. Di ba sabi natin kanina, the colonies are very mucoid. Next. So we perform string tests for Klebsiella. So anong ginagawa dyan? Yung ating inoculating loop, ididikit lang natin siya sa ating colonies and parang iaangat lang natin yung colonies. Kapag yung colonies nakapag-produce siya ng 1 meter long na string, parang presumptive test that the colony is belong for the Klebsiella species. So first we need to discuss the first microorganism that under the Klebsiella. We have Klebsiella granulomatis, so causative agent of Granuloma inguinale. So this microorganism can be transmitted via sexual intercourse because this is a sexually transmitted disease. So the identification of this microorganism does not gram stain or grow on laboratory media. In clinical specimen, right stain or Jamesa stain, ano yung makikita natin pag ginamit natin itong stain na to? Donovan bodies. Since sabi nga natin kanina, hindi siya pwede tumubo sa ating culture media. Anong gagamitin natin? Human monocytes. So these Donovan bodies are intracellular pleomorphic bipolar using bacterial cells. So this is the example of infection that causes by Klebsiella granulomatis. So kung makikita nyo, itong mga to, ito yung mga pinoproduce na clinical significance ni Klebsiella granulomatis. So kaya nga pinopromote natin yung ating safe sex of using condoms during sexual intercourse. So this is the example of Donovan bodies under the microscope. So sabi, clusters of blue or black staining, bipolar chromatin condensations in large mononuclear cells in granulation, tissues infected by Klebsiella granulomatis. So sinabi natin, bipolar staining. Kasi ito yung kanyang both ends. Next. Next, we have Klebsiella pneumoniae. In McConkey agar, encapsulated mucoid colonies are tend to string. So the sputum of the patient infected by Klebsiella pneumoniae is current jelly-like sputum. And the biochemical test for Klebsiella pneumoniae is negative, negative, positive, positive in TSI, A over A with gas, in gelatinase, positive, and in string test, also positive. Next, we have the Klebsiella oxytoca. Sabi natin kanina, Klebsiella species are indole negative except for this microorganism, Klebsiella oxytoca. And next, we have Klebsiella ozonae. So, it causes atrophic rhinitis. So, this is the inflammation of the mucous membrane of the nose. Namamaga yung ating mucous membrane sa ating ilong. So next, we have hapnia. So the empiric reaction for hapnia is negative, variable, positive, negative. Pag sinabi natin variable, it can be positive or negative result. So the identifying characteristics of hapnia is motile, lactose, late lactose fermenters, ONPG positive, LDC positive, ODC positive, and the only organism that represents Hafnia is Hafnia alveoli. Kasi guys, ang tawag kasi kay Hafnia noon is Enterobacter alveoli. So, how to differentiate Hafnia and Serratia? So, the biochemical test for Hafnia is DNA is negative, lipase negative, and gelatinase negative. While in Serratia Majority of the biochemical tests are positive. So kung makikita nyo dito, negative si half niya, whilst Russia is positive. Next. So tatanda natin guys, yung mga microorganism that is positive in Vogue Proskauer or VP. Tandaan natin yung mnemonic na cash. So we have Klebsiella, Enterobacter, Serratia, and half niya. Kapag nakita sa board exam or exam natin na positive sa VP, isipin nyo lang tong apat na microorganism na to. Kasi sila lang yung microorganism na positive sa ating Vogue Proskauer. And guys, tip ko lang, if the microorganism is positive in Vogue Proskauer, the result for metal red dapat negative. Kung ang metal red is positive, dapat ang Vogue Proskauer is negative. Hindi sila pwede na parehas. So, sila ay opposite. Tatandaan natin. Next, we have Enterobacter. So, the empiric reaction for Enterobacter is negative, negative, positive, positive. Kung babalikan natin yung discussion natin kanina, sinong microorganism ang may ganitong laboratory test or biochem test? So, si Klebsiella. So, H2S negative, MR negative, indole negative, VP positive. So, naka-asterisk guys. Tanda natin, Enterobacter is motile, citrate positive, and lactose positive, except for Enterobacter taylori because this is a lactose negative. And guys, baka magtanong, so paano natin ma-differentiate si Klebsiella from Enterobacter? So, na-discuss natin kanina guys, that they have the same biochemical result, which is the negative, negative, positive, positive. Pero, ma-differentiate natin si Enterobacter from Klebsiella, we need to perform motility testing because Enterobacter is motile, while Klebsiella is non-motile. Tatandaan natin, baka lumabas sa board exam. Paano i-differentiate si Enterobacter and Klebsiella? We need to perform motility testing. Again, Enterobacter, motile, Klebsiella, non-motile. Next. So, naka-indicate na rin pala dito yung kanilang different reaction sa ating testing. Next, LDC negative ang ating Enterobacter except for Enterobacter aerogenes and Enterobacter gergoviae because they are LDC positive. Also, Enterobacter species are slow urease producer except for Enterobacter gergoviae because Enterobacter gergoviae is rapid urease producer. Tatanda natin, majority ng Enterobacter species are slow urease producer except kay gergoviae kasi mabilis siya makapag-produce ng urease. Next, we have the Enterobacter cloacae. So this is the most common isolated, so arginine positive and lysine negative. Next, we have Enterobacter aerogenes. This is the second most common isolated Enterobacter. So the biochemical test for arginine is positive while lysine is negative. Kung i-compare natin sila, makikita nyo naman yung kanilang arginine and lysine result. So next, we have the Enterobacter sakazaki. So produces yellow pigment that aids in its presumptive identification. So kapag nakita tayo ng ganitong colony sa ating culture media, presumptive lang natin that is Enterobacter sakazaki. Pag sinabi natin presumptive, hindi pa yun sure kung sa Enterobacter sakazaki pa lang. So we need to perform what? Biochemical testing uh, to confirm if the colony is belong talaga to Enterobacter sakazaki. Next, we have Serratia. So Serratia causes opportunistic infection in patient undergoing chemotherapy and immunosuppressed patient. So the commonly isolated microorganism that under Serratia species is Serratia marcescens. So what is the identifying characteristics of this microorganism? DNA is positive, gelatinase positive, lipase positive, and of course, ONPG positive. So, delayed lactose fermenter. And sabi natin kanina, that Serratia marcescens has enzyme prodiosine, which is responsible for the red pigment of the colonies. So this is an example of Serratia marcescens. Next, we have Pantuweya. So the in-peak reaction for Pantuweya is negative, negative, positive, positive. So the only microorganism that is isolated under Pantuweya species is Pantuweya agglomerans. So nitrate reduction negative produces yellow colonies in the culture media, LDC negative and ODC negative. Next, we have salmonella. The in-peak reaction for salmonella is negative, positive, negative, negative. So the mode of transmission for this microorganism is ingestion of the contaminated food and water. So na-discuss na natin that salmonella produces hydrogen sulfide. So H2S positive, motile, urease negative, and lactose negative. So the infection causes by salmonella species enteric or typhoid fever. So 80% of typhoid fever uh, causes by Salmonella typhi, while 10% of the infection of the typhoid fever causes by Salmonella paratyphi, serotype A and B. Next, we have the bacteremia causes by Salmonella cholera suis and enterocolitis and gastroenteritis causes by Salmonella typhimurium. So, how to assess inside the laboratory kapag merong typhoid fever si patient? In the first week of typhoid, so it produces skybala stool or goat-like dropping. Pag sinabi natin goat-like dropping, ito yung parang dumi ng mga kambing. So, para siya mga bilog-bilog. So, tinatawag natin siya na skybala stool. And the specimen of choice for typhoid fever in the first week of infection is blood. Kasi gagamitin natin siya for blood culture. For second week of infection, this is the colonization stage. So, the bacteria causes rose spots in the patient. So, the specimen of choice is urine and stool. So, baka may magtanong, sir, ano yung rose spots? So, this is the example of rose spots. Nagkakaroon ng parang batik-batik si patient sa kanyang body. That's his rose spots. Next, we in the third week of infection of typhoid fever, this is the convalescent stage. So the specimen of choice, of course, is serum. And we need to perform Widal or typhoid test. So Widal test or typhoid test, this is the test to ascertain the diagnosis of typhoid fever. As with all the serological tests, the rise in antibody levels needed to perform the diagnosis takes 7 to 14 days, which limits its applicability in early diagnosis. Other means of diagnosing Salmonella typhi and paratyphi includes culture of blood, urine, and feces. Kasi guys, di ba sabi natin kanina, in third week, we need to collect serum. Bakit serum? Kasi guys, in vital test, ang check na natin is antibody levels. Kasi nga guys, di ba sabi natin, bakit yung nagkakaroon ng antibody? Nagkakaroon din ng antibody if the patient is exposed to the infection. Nagpo-produce na tayo ng antibody against dun sa infection. So, the principle of this is the antibody in the serum produces in the response of Salmonella organism. The kit contains antigen suspension that are killed bacteria and they were stained to enhance the reading agglutination test. So, ano nangyari dito? Yung kit natin, meron siyang antigen na Salmonella. Since sa serum natin, meron tayong antibody, ano gagawin ng antibody? Si antibody is mag-attach dun sa ating antigen. Di ba? Sa ating immunosero, diniscuss, si antibody a-attach sa antigen and magkakaroon ng reaction. So, magkakaroon ng agglutination sa ating sample. So, the blue stain antigens are specific to the somatic antigen or O antigen, while the red stain antigens are specific to the flagellar antigens or H antigen. So, this is the example of vital test inside the laboratory. So, pinaperform talaga natin siya. So, guys, kung mapapansin nyo, merong agglutination na nangyayari. So, we have positive control and negative control. So, importante yan pag magpa-perform tayo ng ating test inside the laboratory. Next, after the Salmonella, we have Shigella. So, the in-vivo reaction for Shigella is positive, positive, and negative, negative. Other name for Shigella is inert bacilli. Again, kaya natin siya tinawag na inert bacilli kasi majority ng kanyang biochemical tests are negative. So, Shigella are lactose negative, non-motile, causa shigellosis or bacillary dysentery. In TSI, result is K over A or alkaline over acid. The methyl red reaction is positive. And again, majority of the biochemical tests are negative. The incubation period for Shigella is in early stage is 1 to 7 days. In diarrheic stage, 3 days na siya. So the patient produces watery stool or watery diarrhea. And we have dysentery phase. So the virulence factor for Shigella, it has neurotoxin, which cause paralysis and death. It affects the CNS or the central nervous system. So we have different serotypes of Shigella. So we have serogroup A, serogroup B, serogroup C, and serogroup D. In serogroup A, we have Shigella dysenteriae. So the other name for this microorganism is Shigas bacillus. Next, we have S. flexneri. Other name is strong bacillus. Para hindi malito, flex. Flex mo lang ng flex. Strong yan. Flexneri, strong bacillus. Next, serogroup C, which is the Shigella boydii, also known as the Newcastle Manchester bacillus. And of course, the serogroup D, Shigella sonei or the Duval's bacillus. Tanda lang natin, D. Duval's para hindi malito. So, how to differentiate the different serogroups of Shigella? So, we need to perform catalase, ONPG, and mannitol fermentation. So, kung mapapansin nyo, majority of the microorganism is positive in catalase test except for Shigella dysenteriae. And ONPG, positive C, sonei, and in mannitol fermenter, dysenteriae is negative. So, madali naman siya tandaan kasi ginawan ko rin siya ng table. So, babasahin na lang intindihin. So, next, how to differentiate Salmonella and Shigella? So, Salmonella is motile except for Salmonella gallinarum and Salmonella pullorum. So, H2S positive, gas producer, LDC positive, except for Salmonella paratyphi because this microorganism is LDC negative. So, kasi Shigella naman is non-motile, H2S negative, not a gas producer, and LDC negative. Next, we have Proteus. So Proteus is rapid urease producer. So sinabi natin kanina, yung ating mnemonics, ppm. So Proteus is typical swarming motility. Kapag nabasa sa board exam, microorganism that has swarming motility. Wag na mag-isip ng iba-iba. Sagutan natin Proteus. H2S positive in TSI, but not in LIA. Kasi nga, in TSI, we have space. In LIA, we have cyst. Diba? Pakibalikan yung ating culture media topic regarding sa TSI, LIA, McConkey, EMB, balikan. Kasi connected to doon. So next, Proteus is a source of antigen for whale Felix test. So, baka may magtanong, Sir, para sa yung whale Felix test? This test is used for the serological text diagnosis of rickettsia. Tatandaan, whale Felix test is serological test for the detection of rickettsial disease. We have Proteus vulgaris and Proteus mirabilis. So this is the source of OX2 and OX19 antigen, while Proteus mirabilis is the source antigen of OXK or the Kingsbury stain. So, paano natin ma-differentiate si mirabilis and vulgaris? Ito guys, makikinig tayo. In indole test, P. mirabilis is negative, while Proteus vulgaris is positive. Paano natin siya matatandaan under indole test? Vulgar, 'di ba? Parang sa typical, uy, na vulgar siya, nakita, positive. So in indole test, positive C. vulgaris. ODC, positive C. mirabilis, negative C. vulgaris. In chloramphenicol testing or in susceptibility testing, mirabilis is susceptible and vulgaris is resistant. Tatandaan natin yung kanilang mga laboratory tests. Next. And bago pa natin i-move sa next slide, so tandaan lang natin, mirabilis associated in wound infection and UTI, while vulgaris can cause pneumonia and septicemia. So next we have Providencia. So the biochemical test for Providencia is positive positive, negative positive, lactose negative, deaminase positive, H2S negative, motile but not swarming, LDC, ODC, ADH negative, and the commonly isolated Providencia microorganism or species are Providencia ritgeri and Stewarti. So tatandaan lang natin yung urease test, adonitol test, and Trehalose test. So, nakatable na, pakibasa. Next, we have Morganella. The imbic reaction for Morganella is negative, negative, positive, positive. So, Morganella morgani is the only isolated species. So, lactose negative, deaminase positive, H2S negative, LDC negative, urease positive, kasi nga, siya ay under sa ating pneumonics din. And of course, ODC positive. Next, we have Citrobacter. So Citrobacter frundi is the most common isolated species. Resemble on E. coli on McConkey. Kapag tumubo sila sa McConkey, mapagkakamalan siyang E. coli. Pero paano sila ma-differentiate? We need to perform what? Indole and H2S testing. Kasi guys, in H2S, Citrobacter is positive. Kasi nga kasama siya sa ating mnemonics, yung space. While E. coli is negative. Pero daw, in indole test, Citrobacter is negative in indole. While E. coli is positive in indole and negative in H2S. So guys, doon pala magigets nyo na. Paano natin ma-differentiate yung mga microorganisms? So, Citrobacter, napagkakamalan din siyang Salmonella. Pero we need to perform ONPG test and LDC and urease test. Bakit? Salmonella is urease negative. Pero si Citrobacter is urease producer. Pero nga lang, slow urease siya. Nandaan natin yung kanina, C-KISS. For ating slow urease producer. For rapid, balikan lang natin, we have ppm. For Edwardsiella, we have positive, positive, negative, negative biochemical test result. So, the common isolated species under Edwardsiella is Edwardsiella tarda, resembles a Salmonella. Why? Because they are both H2S positive. But in ONPG test, Edwardshella tarda is ONPG negative, pero si Salmonella daw is positive sa ating ONPG testing. Also, Edwardshella tarda is indole positive, citrate negative, while Salmonella is indole negative, citrate positive. So, pakitandaan yung mga ating biochemical testing. So, how to differentiate Edwardshella from E. coli? So, tandaan lang natin, Edwardshella produces hydrogen sulfide while E. coli is negative. Lactose negative si Edwardshella, pero si E. coli is lactose positive. Tandaan, Edwardshella is LDC positive, ODC positive, and ADH negative. Next, we have Yersinia. So we have three pathogenic species under Yersinia species. We have Yersinia pestis, Yersinia enterocolytica, and Yersinia pseudotuberculosis. So first, we have Yersinia pestis causes plague. So guys, kung nagbabasa kayo sa Facebook, di ba sa China, kumakalat na naman daw yung plague disease. So it causes by Yersinia pestis. So this bacteria can be transmitted via fleas. So, considered as potential bioterrorism agent. So, paano din siya natatransmit? Kasi guys, this Yersinia pestis can be isolated in rodents or rats, which is the natural reservoir of this microorganism. So, paano nangyari yung plague? So, the rat carries plague or the bacteria. So, kasi, di ba, sa ating sa rats, it has fleas, drinks the flat, drinks the rat's blood. So, ano nangyari? Bacteria multiplies in fleas abdomen. Kasi, itong fleas na to, pwede tayong makagat. Once na makagat na tayo, the the bacterial infection can be transmitted. So, the human can be infected. Next, we have Yersinia enterocolytica. So, causes enterocolitis in humans. It is acquired by drinking contaminated water or by eating contaminated meat. So, in colony, it produces bullseye colonies. So, ONPG positive because it is delayed lactose fermenter. Sucrose positive. In McConkey, it is colorless at 18 hours. In TSI, it produces A over A or acid over acid. So this is the example of Yersinia enterocolitica. So it has bull's eye appearance. Next, we have Yersinia pseudotuberculosis. It is a rare cause of lymphadenitis in children. So guys, mali lang ako ng spelling. So that is Yersinia pseudotuberculosis. So what is the unique characteristic or identifying characteristic of Yersinia? So in McConkey, small coccobacilli that produces small pinpoint colonies suspected if TSI is yellow or red or acid, alkaline, or A over K. So all are motile in 37 degrees Celsius except for Yersinia pestis. Kasi nga, di ba, meron tayong mnemonics na sky. So what is selective media for Yersinia? We have the CIN agar or the self-soludine ergasa novobicin agar. This CIN agar also used for the isolation or selective culture media for Aeromonas and Plesiomonas. This culture media contains mannitol, peptones, yeast, neutral red indicator, and crystal violet. So guys, tinable ko na rin siya. The microorganism, the disease associated, the sucrose test, rhamnose test, and sorbitol test. So, pakibasa, pakiintindi. Next, we have Haemophilus. So guys, familiar na tayo dito kasi na-discuss na natin siya sa previous topic sa ating culture media. So, didiscuss na natin siya para maintindihan natin siya ng buo. So, Haemophilus, this is the most species are normal flora of our respiratory tract or upper respiratory tract. So, Haemophilus species, especially Haemophilus aprophilus, are considered member of HASEC or the Haemophilus actinobacillus actinosetem comitans, Cardiobacterium hominis, Echinella cordens, and Kingella group. So, sabi nga natin dati, this HASEC microorganism causes endocarditis. Tatandaan, this microorganism causes endocarditis. And Haemophilus is pleomorphic gram-negative coccobacilli ranging from small to filamentous. So growth requirement for Haemophilus, we need hemine and NAD. So hemine is X factor which released from hemoglobin and NAD or V factor which is the hate label compound. This NAD compound also seen in S or Us because S or Us produces V factor which is essential for the growth of Haemophilus. Kaya kung mapapansin nyo, si Haemophilus pwede siya tumubo sa colony ni S. aureus o nagkakaroon tayo ng Haemophilus satelitisium sa culture media. Na-discuss na rin natin itong culture media for Haemophilus. It will not grow on sheep's blood agar because SBA contains NADAs which inhibit the production of NAD. So, pwede tayong gumamit ng blood agar plate if the blood contains of horse or rabbit. Tanda natin, pwede tayong gumamit ng blood agar if the blood is horse or rabbit's blood. Kapag sheep's blood, hindi pwede. Kasi nga sabi nga natin that sheep's blood contains NADAs. Ano ginagawa ulit ng NADAs? Inactivates NAD. Tatandaan. So, in routinely laboratory setup, we use chocolate agar plate. So, this is the ideal culture media. Grows at 35 to 37 degrees Celsius with 5 to 10% carbon dioxide and susceptible to drying and temperature changes. So ano siya? Sensitive siya sa pabago-bago ng temperature, madali siya mamatay. So the colony for Haemophilus is smooth, round, flat, opaque, and tan on chocolate agar plate. So this is the example of Haemophilus under or on chocolate agar plate. So, hindi ko na siya babasahin kasi na-discuss natin si Haemophilus satelitisium. So, what is the unique characteristic of Haemophilus? Non-motile, catalyzed positive, and oxidase positive. So, first we have the Haemophilus influenzae, formerly called Pfeiffer's bacillus or Bacillus influenzae. It it causes major Haemophilus that causes meningitis in children. And Haemophilus influenzae type B or HIV vaccine has greatly reduced childhood meningitis and other invasive diseases caused by this serotype. Kaya pa lang nung bata tayo, nag-i-inject na tayo ng vaccine na to para di tayo ma-infect ng Haemophilus influenzae. So, Haemophilus influenzae isolate should be tested for beta lactamase. So the specimen of choice for Haemophilus influenzae is blood, sputum, CSF, and of course, pwede pala daw, eye swab. Tatandaan natin, eye swab. In latex agglutination, specific detection for HIV capsular antigen. Next, we have Haemophilus aegypticus. So causes pink eye, a very contagious conjunctivitis similar to H. influenzae with the exception of being sucrose positive. Kasi si influenzae is sucrose negative. So tanda natin, pink eye. Next, we have Haemophilus influenzae by group Egyptius. So causes conjunctivitis caused by invasive disease called as Brazilian purpuric fever. Tatanda natin, Brazilian purpuric fever, Haemophilus influenzae by group Egyptius. Next, we have Haemophilus ducreyi, causes genital ulcer, can be transmitted via sexual intercourse, produces chancroid and buboes. So, namamaga yung ating lymph nodes. So, in chocolate agar plate with vancomycin is used to inhibit the normal flora and contaminant. Di ba sabi natin kanina, chocolate agar plate ang ating ginagamit na culture media for the isolation of Haemophilus. Pero gagamit din tayo ng vancomycin. So this is the example of chancroid in male genital. So guys, in summary, this is the different microorganism of Haemophilus. We have Influenzae, Ducreyi, Egyptius, Aprophilus, Haemolyticus, Parainfluenzae, para, Paraprophilus. So, di ba sabi natin kanina, Haemophilus required X factor, V factor, horse blood, or ALA. So, pakibasa, intindihin kung ano yung mga microorganism na kailangan ba niya ng V factor, X factor para tumubo. For example, Haemophilus aprophilus, hindi niya kailangan ng X factor, V factor, and horse blood. Pero need niya ng ALA. ALA is amino-levulinic acid. So ano to? Is converted to porphyrin. So, nakatable na siya, madali na siya intindihin, kasi ito yung pinaka-summarize ni Haemophilus species. I hope may natutunan kayo sa lecture. So if you have any concerns and clarification, don't hesitate to message me as especially sa Med 182. So if you have any concern, don't hesitate to message me in Teams. So stay safe and thank you so much for listening.